Hello students, today again we are going to continue with same biology chapter number 4, molecular basis of inheritance. Okay, in last lecture we have covered the topic replication of DNA and mechanism, or we can say process of DNA replication. When we say DNA replication, we have studied at the end that the replication of DNA is semi-conservative. Okay, so today we are going to prove it with the help of an experiment. That means today we are going to study experimental confirmation of semi-conservative replication. Let me revise you. What do you mean by semi-conservative replication? In newly formed DNA molecule, one strand is old and other strand is newly synthesized. This is called semi-conservative replication. After the process of replication, that means when the replication takes place, one strand is new and other strand is old. This is called semi-conservative. Semi means half. Okay, and half strand is old, half strand is conserved. So that means conserved. Okay, and half is newly constructed. So it is called as semi-conservative replication. This we are going to study with the help of one experiment today. Michelson and Stahl in 1958 performed experiment to prove that replication of DNA is semi-conservative by using equilibrium density gradient centrifugation technique. Now, when we say equilibrium density gradient centrifugation technique, I will discuss this in detail. But before that, let us see what was the experiment that Michelson and Stahl have performed. Michelson and Stahl have performed an experiment in which he have used two mediums, that is N15 medium, N14 medium. Okay, and this in N15 as well as N14 medium, the replication process was seen after every 20 minutes because the reproductive cycle of the bacteria that was used here, that is E. coli bacteria, have 20 minutes of its replication cycle. So after every 20 minutes it replicates, that is it doubles. So let us start now. In first, N15 medium was used. In this N15 medium, E. coli cells were grown. Okay, in this E. coli cells with N15 medium, that is heavy isotope of nitrogen was used and E. coli bacteria was grown. What was the result? After few generations, all nitrogen bases will contain same N15 isotope in DNA of E. coli cells. Okay, then after some time, this E. coli bacteria from N15 medium was transferred to N14 medium. And what was seen there? Okay, after the transfer, E. coli was transferred to medium containing N14, that is light isotope, and the cell samples were analyzed after replication cycle by density gradient centrifugation technique. Replication cycle, I have told you, it means every 20 minutes. Replication cycle consists of 20 minutes. Okay, and now we will discuss the technique, that is density gradient centrifugation technique. Okay, in this technique, samples are separated using density gradient centrifugation. Density means weight, gradient means difference, and centrifugation is a process in which the samples are rotated at a very high speed. Okay, centrifuge machine is there. In that, samples are kept in a test tube and the test tubes are spin at a very high speed, and because of that the samples are separated in the test tube, and this is performed with the help of heavy metal salt like cesium chloride. Okay, now let us see the observation part. What will happen when it was in N15 medium? The result was that all the DNA was showing N15 isotope, and with the help of centrifugation technique, the band was obtained. Okay, centrifugation technique. After the centrifugation technique, the band was obtained at the bottom of the test tube. You can see here, since the N15 is a heavy isotope and it is having high density, so the band is formed at the bottom of the test tube. You can see here at the bottom of the test tube, N15 medium. After 20 minutes, we will be able to see the first generation. Now what we see after first replication is that DNA was neither heavy nor light. That means it does not show purely N15 nor purely N14, but in between it shows the hybrid N14 and N15 DNA molecule that can be observed by centrifugation technique with the help of band that is present in between the test tube. Only N15, the band was at the bottom of the test tube, and when hybrid was obtained, what was happened, that the band was obtained in between the test tube. So see here, after the first replication technique, what was happened, the two DNA molecules were formed, in which one was the old strand. You can see here, this is the old strand, and the other strand is newly constructed. Okay, so this indicates that the replication is semi-conservative. We can see still in further generation, that is in generation 2, which will be happening after 40 minutes, because first generation was after 20 minutes, so second generation will take place after 40 minutes. 20 plus 20, each cycle is of 20 minutes, because E. coli bacteria multiplies in every 20 minutes. Okay, so the DNA that will be present in E. coli bacteria will also multiply in 20 minutes. So same thing here, if after generation 1 we are going to get 4 molecules of DNA, in which each strand in one DNA molecule will be old, okay, and the other will be newly constructed. Here one old and the other one is newly constructed. Same, one old, one newly constructed, one old and one newly constructed. So total 4 DNA molecules are formed, in which we can say that 2 types of bands were obtained. Okay, so after second generation, 2 density gradient bands were obtained, one at N14, N15, that is at hybrid position, and the other one was at N14, that is it is having a light weight, so it is uppermost part of the band. Okay, the band is formed at the top of the test tube, so we are getting hybrid as well as light. Okay, so we can give the conclusion from this that the position of the bands after 2 generations clearly prove that DNA replication is semi-conservative. Thus, at every generation of the parental DNA becomes 50%. So, in the next generation, it will reduce to 25%, then it will get reduced to 12.5%, then 6.25%. So, at every generation, we are going to get 50% less of the parent generation. Okay, thank you.